In 1911, a virologist named Francis Rouse was studying the development of fibrosarcomas in chickens. Much to his surprise, he discovered that when the cellular extract of one chicken's sarcoma was removed and injected into the system of a healthy chicken, that healthy chicken also went on to develop the same form of tumor from which the cellular extract came. This was a significant discovery as it was the first time that cancer was seen to have a viral property. An analysis of the tumor extract found that there was a virus expressing high levels of one unknown protein. It was concluded that the expression of this protein was what was causing the tumor growth. Many years later, this gene was identified in humans as SRC and its human protein product as c-SRC, short for cellular SRC. The SRC protein is a widely expressed protein tyrosine kinase, found to some extent in most human cell types. As a tyrosine kinase, SRC acts on its substrates by phosphorylation of tyrosine residues as part of the activation of those substrates' signal transduction cascades. With over 50 different substrates, SRC is able to play a role in a wide variety of cellular functions, many involved in the regulation of cell growth. In normal cells, SRC is generally found in its inactive form. The exception to this is when SRC is needed to initiate signaling pathways or in cases of large-scale or rapid growth, such as in tumor cells. In order to better understand how SRC could be a target for anti-cancer therapies, it is essential to take a closer look at its structure. At the N-terminus, the SRC protein contains a myristoyl group, which is a 14-carbon group that directs the protein to the cell membrane where it is needed to carry out its kinase function. Next is a unique domain, which is the least conserved region in an otherwise highly conserved protein. Following that are the SH3 and SH2 regulatory domains, an SH2 kinase domain linker, and the bilobed SH1 or kinase domain, which contains two key regions, the alpha-C helix and the activation loop. Also on the activation loop is tyrosine 416, a key regulatory tyrosine. This is followed by the C-terminal domain, which contains the second regulatory tyrosine. The way that each of these protein regions interact allows for highly specific enzymatic activity and regulation. Specifically, SRC is able to phosphorylate its substrates at the targeted tyrosines through the use of the large and small lobe of its kinase domain. Between the two lobes of the kinase domain is a cleft made usable as an active site for phosphorylation by the favorable interactions of the inward orientation of the alpha-C helix. With the alpha-C helix in, SRC is ready for ATP binding and subsequent tyrosine kinase activity. To stabilize ATP in this cleft, small lobe glycine residues interact with the phosphates on ATP, while two other strands in the small lobe interact with the adenosine portion of the ATP. To properly position the substrate so that its correct tyrosine is phosphorylated, an interaction between the activation loop and the substrate's target tyrosine occurs. With the ATP in the substrate now in position, an aspartate residue in the kinase domain facilitates the nucleophilic attack of the substrate's tyrosine R group on one of the ATP phosphates, ultimately leading to the phosphorylation of that tyrosine and the continuation of that substrate's specific signal transduction cascade. Since it is not always favorable for the cell to promote these cascades, the activity of SRC needs to be tightly regulated. Such regulation is achieved indirectly through conformational changes of SRC, brought on by the phosphorylation and dephosphorylation at one of either of the two key tyrosine residues. In vivo, phosphorylation of 416 generally leads to increased or full activity, while phosphorylation of tyrosine 527 leads to downregulated or completely inactive SRC. The transition observed between these two states is described as the latch, clamp, and switch model of SRC regulation. In the inactive state, both the latch and clamp states are observed. When SRC is latched, the SH2 domain is bound to the phosphorylated tyrosine 527, bringing it into loose contact with the large lobe of the kinase domain. SH3 is bound to the SH2 kinase linker, bringing it closer to the small lobe. With this configuration, the clamping of SH2 and SH3 to the back of the kinase domain is possible. With this binding, the alpha-C helix is made to point outwards, disrupting the activity of the activation site and its ability to bind ATP. In order to become fully active, the SRC protein must undergo unlatching, unclamping, and a switch. Unlatching and unclamping is achieved through dephosphorylation of tyrosine 527. This disrupts the binding with the SH2 domain and leads to the unclamping of the SH2 from the large kinase lobe. With the SH2 now unbound from both tyrosine 527 and the kinase domain, the activation loop tyrosine 416 is phosphorylated, switching the activation loop from an inactive conformation to an active conformation. The SH3 domain is simultaneously unclamped from the small kinase lobe, and SRC has reached its full activation state. having both the alpha helix oriented inwards and the activation loop in an active state. This process can begin again by the dephosphorylation of tyrosine 416 by protein phosphatases and the phosphorylation of tyrosine 527 by protein kinases, allowing SH2 to latch once again to tyrosine 527 and the rest of the mechanism to follow. More often than not, SRC is not needed in large quantities in the cell, and so it generally exists in its deactivated state. However, when issues arise with any of SRC's regulators or if components of the deactivated complex are mutated, the cell loses control over SRC and many of the oncogenic pathways it promotes. Active SRC and its regulators have been found in significantly elevated amounts in a diverse group of cancers, leading many scientists to look at the benefits of targeting SRC as a way to halt tumorigenesis. One effective SRC-inhibiting drug, dasatinib, is used to combat chronic phase leukemia via competitive inhibition of SRC's active site. This prevents ATP from binding and downregulates the effects of SRC signal transduction cascades. Many other anti-SRC therapies are still in the early stages, but with a protein as globally effective and intricately controlled as SRC, the options and benefits of targeting therapies are innumerable. This one protein is already leading to some truly revolutionary discoveries, and it can all be traced back to the work of one scientist and his chickens over 100 years ago.